Revisiting the coelacanth, a living fossil's secrets. Imagine a fish that has remained virtually unchanged for 65 million years. A true living fossil that continues to surprise scientists with new insights into vertebrate evolution. This is the coelacanth, a fish that has been extensively studied, but still holds secrets that can reshape our understanding of how vertebrate skulls evolved. A groundbreaking study published in the journal Science Advances by researchers from the University of Sao Paulo, USP, in Brazil, and the Smithsonian Institution in the United States has done just that. The study delved into the cranial musculature of the African coelacanth, Latimeria chalumnae, and uncovered some astonishing findings. It turns out that only 13% of the previously identified evolutionary muscle novelties for the largest vertebrate lineages were accurate. The researchers also identified 9 new evolutionary transformations related to innovations in feeding and respiration in these groups. This revelation is significant because it shows that the coelacanth is even more similar to cartilaginous fish like sharks, rays, and chimeras, and tetrapods, including birds, mammals, amphibians, and reptiles, than previously thought. But here's where it gets controversial. Unquote. The study also found that the coelacanth is more distinct from ray-finned fish, which make up about half of living vertebrates. One of the most surprising discoveries was that some muscles previously thought to be present in coelacanths were actually ligaments, which are structures incapable of contraction. These muscles, responsible for actively expanding the buccopharyngeal cavity, the area from the mouth to the pharynx, are crucial for food capture and respiration. The study showed that these supposed muscles in coelacanths were actually ligaments, which means they do not contribute to movement, but rather transmit it. Ray-finned fish, Actinopterygii, and lobe-finned fish, Sarcopterygii, diverged from a common ancestor approximately 420 million years ago. The Sarcopterygii include fish such as coelacanths and lungfish, as well as all other tetrapods, because they evolved from an aquatic ancestor. These include mammals, birds, reptiles, and amphibians. In ray-finned fish, such as aquarium carp, it is easy to see how the mouth moves to suck in food. This ability gave Actinopterygii a significant evolutionary advantage. Today, they comprise about half of all living vertebrates. This is a fundamental difference from other fish, such as coelacanths and sharks, which primarily feed by biting their prey. And this is the part most people miss. In previous studies, it was assumed that this set of muscles that would give greater suction capacity was also present in coelacanths, and therefore would have evolved in the common ancestor of bony vertebrates. However, the new study shows that this is not true. This only appeared at least 30 million years later in the common ancestor of living ray-finned fish. Coelacanths are extremely rare fish that live about 300 meters below the surface of the water and spend their days in underwater caves. One reason they have changed so little since the extinction of the dinosaurs is that they have few predators and live in a relatively protected environment. This has resulted in slow changes to their genome, as shown by a 2013 study published in the journal Nature. Coelacanths were first known only from fossils from about 400 million years ago. It was not until 1938 that a living animal was discovered, much to the astonishment of scientists. In 1999, another species, Latimeria chalumnae, was discovered in Asian waters. Due to the rarity of specimens in museums, researchers from USP and the Smithsonian Institution's National Museum of Natural History had to persevere to find an institution willing to lend animals for dissection. The Field Museum in Chicago and the Virginia Institute of Marine Science, both in the United States, finally agreed to lend one specimen each. According to Dotavo, G. David Johnson, co-author of the article, deserves credit for obtaining the loan. Johnson, born in 1945, was probably the greatest fish anatomist of his time, according to Dodavo. He died in November 2024 after a domestic accident while the study was under review. Contrary to what it may seem, dissecting a specimen does not mean destroying it as long as it's done properly, says Dodavo. The researcher, who has been conducting this type of study for over 20 years, spent 6 months separating all the muscles and skull bones of the coelacanth. These structures are now preserved and can be studied individually by other scientists, eliminating the need to dissect a new animal. Seeing each muscle and nerve firsthand allowed the authors to identify what was actually in the coelacanth's head with certainty, point out previously undescribed structures, and correct errors that had been repeated in the scientific literature for over 70 years. There were many contradictions in the literature. When we finally got to examine the specimens, we detected more errors than we'd imagined. With this information, the researcher used three-dimensional microtomography images of the skulls of other groups of fish, both extinct and living. These images are made available by other researchers who study fish anatomy when they perform 3D scans. From images of the skull bones of other fish from completely extinct lineages, Dodavo and Johnson were able to infer where the muscles found in coelacanths would fit, elucidating the evolution of these muscles in the first jawed vertebrates. In future work, Dodavo intends to analyze similarities with the muscles of tetrapods, such as amphibians and reptiles. So what do you think? Does this new information challenge your understanding of vertebrate evolution? Do you agree with the findings, or do you have a different perspective? Let's discuss in the comments.